Howdy, today I'm looking at genes, introns, and exons. Thanks to Sam Moyle for producing this PowerPoint. So the first understanding we're going to look at today, a gene consists of a unique sequence of nucleotides that codes for a functional protein or RNA molecule. Distinguish between exons and introns as coding and non-coding segments of DNA found in eukaryotes. And describe how both exons and introns are transcribed, but only information from exons is translated. So we're going to use a bit of an analogy for what we're going to see today. So imagine a recipe book. So there's chapters, sweets, cakes, jellies, savoury soups, casseroles, and so on. And within each chapter, there's individual recipes: chocolate cake, orange cake, vanilla cake, chicken, sweet corn soup, and so on. The whole genome is like the recipe book. Chromosomes are the chapters that contain the recipes, and then the genes are the recipes that code for the formation of a particular thing. So a recipe that tells you how to make a cake, for example, is similar to a gene that codes for the production of a protein or an RNA molecule. Here's my recipe book. Here are my chapters, and those are our chromosomes. Within our chapters, we're going to have particular recipes, and those are the genes on our chromosomes that code for the production of a particular polypeptide, which is another word for protein, or an RNA molecule, depending on what the gene is coding for. And that's the same as a recipe, so a type of soup or a type of dessert. So let's get our terminology. A gene is a sequence of DNA that codes for the production of a protein or an RNA molecule, depending on what the gene is coding for. Why is this important? So DNA is so important because those proteins are very important. They do several jobs. Some of them are structural, so that means that they make up structures inside cells and organisms. But many of them are enzymes, and enzymes are little proteins that speed up chemical reactions. Since life is a whole series of chemical reactions, those chemical reactions are moderated by enzymes. So enzymes can speed up reactions that normally would take a very long time to occur. Some proteins slow down, so some enzymes slow down reactions, but most of the ones we look at, we're thinking of speeding up. So enzymes are very important. So your DNA makes proteins. Those proteins are often enzymes. Those enzymes speed up chemical reactions. If those enzymes don't work, then you lead to a bit of trouble. So I've got some pictures showing what's going on. So here we have a sequence of DNA. So we talked about A, C's, T's, and G's previously. You get a sequence of those A, C, T's, and G's making up your chromosomes. A section of those chromosomes make up a gene, and that gene codes for the production of a protein. So here we have our DNA sequence, which leads to a sequence of amino acids, and we're going to look at how that works a bit later on. Those amino acids then fold themselves into the 3D structure of the protein, and then that protein does a particular job. Up here, we've got a similar picture. So we've got our DNA. A section of that DNA is coding for, in this case, coding for a protein, but over here it can just code for RNA as well. Most of the time we think of genes as coding just for the production of proteins, and they might be enzymes, but also RNA can be produced from genes as well. Over here we have a picture showing what an enzyme does, and we're going to go through this in detail later as well. Here's our gene. It leads to an enzyme, and that enzyme turns substrates into products. So in biology we don't say reactants. In chemistry we say reactants, but in biology we say substrates. And you produce products. So the enzyme takes your reactants and turns them into products. The way it does that is by reducing activation energy. We're going to look through that a bit later on as well. The chromosomes contain your recipes, which are your genes, and on any one chromosome you can have thousands of genes. Some chromosomes are smaller than others, so they have fewer. There's only about 20,000 genes in total in a human genome. Each gene has a specific location on a chromosome, and we call that a locus. You have many loci, you have one single locus. So we can talk about cystic fibrosis, so that's found on chromosome 7. So here we have a picture of chromosome 7. We can say this gene here is found here on the chromosome, and that's where you find your cystic fibrosis gene. Huntington's disease, which is a degenerative neurological disorder, that's found on chromosome 4. So here's chromosome 4, and we know exactly where it is, and the way we figure it out is kind of complicated. So genes have specific locations on chromosomes. Now we need to learn some new words, exons and introns. So all the DNA in a gene is turned into mRNA, but some of the mRNA isn't required to make your final protein. So it's not needed to make your final protein. The parts of a gene that are turned into the protein, we call them exons. And the non-coding parts of the gene are called introns. I don't know an easy way to remember the difference between those. I often get them confused in my head because I think intro, would be included, but it's the opposite of that. and exons, you might think they'd be excluded, but it's the opposite of that. So if you think of in terms of opposites, that might be working for you in terms of remembering the difference between an exon and an intron. Exons code for bits that are turned into protein, introns aren't. So here we have our genes. This is our whole gene that might be leading to our protein that we're going to produce later on. And we can see that gene is made of a section of exons, introns, and exons. So the RNA that you get out is all transcribed. And we'll talk about what transcription is a bit later on, but you turn the DNA into RNA. So all of the gene gets turned into RNA, but then little bits, the introns get cut out. The introns get cut out of the mRNA, just leading to the exons being joined together. And then these exons that are all joined together, they make up the mature RNA, which is then turned into the protein through translation. And I'll have a look at how that works a bit later on. So some key things. The mRNA includes the introns, even though they don't help to make the final protein. The introns get removed before translation. So before the mRNA is turned into protein, or coded for protein, I should say, all the introns are removed and then the exons are spliced together. Now, it's a very complex process that at least that's happening, and I'll show you this video in class. But the key thing is we have our gene, we have our introns that aren't useful. The DNA gets turned into mRNA, so that's our transcription process. The introns get cut out, and then the exons get joined together, leading to the mRNA being produced, and then that's translated and turned into the final protein. So our check. A gene consists of a unique sequence of nucleotides that codes for the production of a protein or an RNA molecule. You need to know the difference between exons and introns, as exons are coding, introns are non-coding. And you need to talk about how both exons and introns are transcribed, but only the information from exons is used to make your polypeptide, your final protein. Today in Flippin Science, we looked at genes, exons, and introns. That's it, Flippin Science today. See ya.